Thank you for the introduction. And today I'm going to talk about this title because I was told earlier on that the theme of this symposium was proteins in medicine. I'm such an obedient person. I decided to, I don't know, change my sort of way of talking this title, okay? So, okay, I got this one. Why? Doesn't move. Well, I have to do it here. Oh, that was switched off somewhere? Oh, something wrong. Anyway, I'm going to talk about, first is Alzheimer's disease, and the next one is mechanical manipulation and the body handliness. But we have Christopherson. He is an expert of this amyloid, so I decided not to talk a lot about it. Okay, something is not working, so, oh, it's working. So this is the amyloid, and just I want to say very quickly is, if it's A beta is one of the proteins or peptide, and if it's A beta 40 and A beta 42, it's normally 10 to 1. But you have more A beta 42, it's just for the Alzheimer's disease patient. And what is 140, 142? This is this. If this is the beta secretase, this is a transmembrane protein called amyloid precursor protein. If first it's cut here and this one gamma secretase, if it's cut here, this is a normal 40 peptides. But sometimes it cleaves 2 amino acid base residue down to the C terminus, then it ends up 142, 42 peptide. And, oops. And it says, if this is the presignaline, that is the active subunit of gamma secretase, and if it has open form, it produces more A beta 40, but if it's a closed form, it produces A beta 42. So some conformation change seems to be important, and this is a bit difficult to read, but it's the dominant inherited. This is forms of Alzheimer's disease, and you can see there's some missense mutation in the APP, the precursor, or the presignaline 1 or 2, that increases the level of A beta 42. And this one, non-dominant forms of Alzheimer's disease, or including sporadic one, also this has the increase of A beta 42. So, and this is some point mutation, I'm not going to detail, and if you can stop the conformation change by these GMS, gamma secretase modulator, then you may be able to stop the conformational change which produces A beta 42. So this is not my work, but I was interested, why? Only 2 amino acid residue difference makes such a big difference in Alzheimer's disease, or no problem. So I wanted to understand the difference in A beta 40, A beta 42 structures. So to do that, we need a chiroptica spectrophotometer. So chiroptica spectroscopy provides vital information on the structures and the dynamics of proteins and the peptide in the condensed phase. However, solid-state samples cannot be generally measured on commercially available instruments due to intrinsic and strong macroscopic anisotropies, which really produce a serious problem. So what I did, I'm not going into detail, but this is the one. We want to measure CD, and you normally detecting this 50 kilohertz signal. There are many, many other factors, but you want ideal electronically and also optically perfect instrument. You aligned it nicely. Still, you end up with this equation. Now, this is the one you are observing. circular dichroism, this is what you want to measure, but linear dichroism and linear birefringence, LD, LB, these terms are normally zero if it's a completely homogeneous system, like in solution, or completely powder-like one. However, because this condensed phase has some orientations, therefore, this LD or LB is much larger, sometimes 100 times, 500 times larger than these CD signals. So you have to be very careful about it. So I try hard even to make instrument which you can measure CD. And we call this universal chiroptical spectral photometers because we can measure not only CD, but LD, linear dichroism, linear birefringence, because these also have a very important information. It's not a rubbish. We can extract information. Therefore, we measure all these, but I just already show you about the CD. And this is what is USCS1 doesn't mean anything to you, so just going to skip it. And this is the second machine, and this is very unique because you have two detectors. It's very strange. Why? Because I wanted to measure the change of phase of some liquid. Yesterday, Penira talked, if it's in solution, the very weak solution, there's no protein-protein interactions, but it's getting more condensed phase, you get the protein-protein interactions. That is important. Remember that. It is. So we try to put the droplets, and you cannot put the normal instrument because it just gravity, it goes down. But in my one, you can put samples horizontally, and then it can evaporate, it gets more and more concentrated, then you may be able to something. So that's one. And another one here is diffuse reflectance CD, so you can just put powders and you can measure CD. So that's what UCS2, and that's just to show you the beta. Now this is 140. Right-hand side is 142. In alpha helix, this is solution state. You have 17% alpha helix and beta sheet. Beta sheet is the one which produces plaques. End up in the Alzheimer's disease, if it's not lucky. And if it is in the Karst film, its alpha helix goes into beta sheet. This is you can measure with our machine. And if it goes to Langmuir-Brogett, also it is just like this more beta sheet. What happens? 142. We'll be surprised to see already in solution, just you had 2 extra amino acid residue in the C terminus. It has more beta sheet. Okay, so this is like this. So that you can find like this. So this is the one, and this is the one you place samples horizontally, and this is the time course. This is the starting solution, this is time, and you're just measuring at 230 nanometer signal, and this is end up in film, so this is getting evaporation. Here, this red one corresponds to this red one. This is the wavelength, normal circular dichroism spectrum. And this green one is this. So in very short period of time, it switches from alpha helix to beta sheet. That sort of thing you can do. And we worked on alpha-synuclein for the Parkinson's disease or some insulins. And you can work on many neurodegenerative diseases like this. And also, this is the G quadruplex, with DNA, normally has 2 strands wind together to form double helix. But at the end of my chromosome, for example, there are 4 strands of DNA forms G quartet. So, this is the one, and this is inside, tethered inside a hydrogel. This hydrogel is made specially to respond to temperature. So if temperature is higher, it just shrinks. And while you're doing it, you change the conformation of G quartet. So this is it. And you're looking at the DNA structure, and you can have this, and it is actually, you can do it repeatedly. So this is one nice thing. And also, this is the hornet silk. And this is the film, already in the film state, but if exposed to steam 90 seconds, it changed alpha helix to coiled coil, and 142nd, it changed to beta sheet. So these things you can measure on the sort of solid state CD, and it's really fascinating. However, beta, we have to wait for Christopherson's talk. So let's go to the main topic, mirror image animals. Isn't it strange? Mirror image animals? Now, there's a midline, I had a mirror here, then I was made up of left-handed, right-handed, you can hold it like this. But internal organs are different. You can see the lungs are actually not the mirror images each other, these slightly different. Heart is only one and the left-hand side, gut coiled in one direction. So we are not bilateral internally. And that is determined by genes. So that is the fascinating part. But there's always an exception. The exception is really exciting. This is a flatfish. Yes, it's a chirality. And this beautiful shell, this is I picked up myself. This is a chiral, externally as well. And this is done by genetically. And this is actually the snail I've been working on. Why snails? Actually, I was trained as a physical chemist. I did some molecular biology, but it's really for me to be adventurous to go into developmental biology. I don't know how to rear snails, but anyway, I fell in love with this one. So, why snails? Why in particular the one I'm working on, Limnea stagnalis it's called? I thought Limnea may become one of the best model animals for studying the organismal morphology, just like C. elegans and Xenopas, everyone using it. And I'm not going into that one. I started working on this, and it was very, very hard. But anyway, I think you agree with me after my talk. Both dextro and sinistro species are found in the wild, and dextro is dominant. And the asymmetry is displayed both externally, that as you saw it. Okay? And secondly, snail handles depends on the direction of spiral cleavage. This is reported 1894, just like this. This is 1927's paper. Even I wasn't born, all right? And one cell stage, fertilized egg, 2 cell, 4 cell, 8 cell stage, and if it rotates this way, at 8 cell stage becomes sinistral one, and if it rotates that way, at 8 cell stage, 48 cell stage, become dextral one. This has been reported. Third, it's hermaphrodite. So it can be either father or mother. Okay? Hence, it both self-cross and cross occur. 4, body asymmetry follows a maternal inheritance. That means even your gene has a left-handed coiling, but if your mother is right-handed, you are born with a body of right-handed. So it's delayed inheritance. And it's determined by a single gene. That was reported in 1923 to 1930 by Sturbent and Boycott-Diver. That is long before the discovery of Watson-Crick and Wilkins' DNA. That means that's really the Mendelian gene. However, we proved that is a single gene locus about this time, and then it was the single gene. At first we determined, and we published this last year. So it's single gene really determined the coiling of the snail. And in many textbooks, it's shown like this. Here, it's a mirror, and fertilized egg, 2 cell, 4 cell, 8 cell, 24 cells. This all embryology development is mirror image of each other. But I realize that is not the case if you look at closely at the particular stage, and that is the crucial stage for determining handedness. Now, this is it. Can you see here? This is left-handed one, and this becomes a right-handed coiling snail. And this, if you look at it carefully, there's a slight difference. So, I will show you more by staining the F-actin and microtubules. The top one is a dextral one, and this is the step when we come from 4-cell to 8-cell stage that we call the third cleavage. And this is a spiral cleavage. And here, at the metaphase, anaphase, already you can see the deformation of cell. So it's called this spiral deformation. And the spindle in red, that is tilted in helical way already. So we name this spindle inclination. Then, at this telophase, this smaller cell is bud off helically. This is the right-hand way in this species. Then you get right-hand snare. In contrast, for this sinister one, there's no chirality at this stage, and the spindle is almost vertical and radially. So there's no chirality, and small cells are formed just right above. And strangely, after that, when it cleaves off, it turns left-handed way. Then it becomes sinister snare. So we thought this must be something really exciting and interesting. So in many textbooks, it shows, but this is the true thing for the sinister one, there's no SD, no SI. And then it becomes sort of mirror image. And if you work on the fascia acuta, that is a sinister only one, so this is a dominant one. Then this is SDSI, but in a mirror image way, because it's much smaller and it becomes like this. So this is a mirror symmetry relation, but there's no mirror symmetry relation, and this part is actually controlled by the single-handedness determining gene. And it started, but you bring in phase with this, you can see it. Okay, the dextral one, and we stain this with actin polymerization inhibition. There is no actin, because we want to know spindle is pushing the cell, or because the cell deformed and the spindle tilted that way, which is fast. But we can clearly show, thank you, sorted out. Now, we can clearly show there is no actin because actin polymerization was inhibited, and there's no inclination of spindle. For this, we inhibited microtubule polymerization. So there's no spindles formed, but you can see a beautiful spiral deformation. So actin is the first, then spindle tilted. So you can find this, and of course you won't see any difference, but it's as expected. So then I go into very strange thing. This is a very classical thing, and only people like me don't know very much about developmental biology, just start doing these things. Now, we start this pure dexter snail and pure sinister snail. Now you can, this right circle is actually the handedness determining gene. Now you cross it, you end up the F1, so 50% dexter, 50% sinister gene. Then you cross this with the pure sinister one, you end up F2. F2 contains 25% gene from dexter one and 75% sinister one on average. And if you inherit the handedness determining gene within that 25%, just like this, of course the body is left-handed because mother is left-handed. However, the next generation, if it is the case, you can produce offspring of right-handed coiling, and if you don't inherit it, it produces offspring, next generation of left-handed coiling. So you have to wait until it produces the next generation. That's time-taking. It's terrible. Quite often they just eat food and don't produce eggs at all. So it's very not good. But anyway, you keep doing it. And at the F6 stage, it's 1.6% dextral and 98.4% sinister. But still, if you inherit that handedness determined gene, you you produce the next generation. So we published a paper in the Nature and Current Biology using this stage, but eventually I managed to make congenic one. Okay, so this is 0.1% dextro gene and 99.9% sinister gene on average. And within that 0.1%, if you have inherited the Handes determining gene, that is F10D, it produces 100%, it does spiral deformation, spindle inclination, and it becomes dexter snail. If it doesn't inherit that one, there's no SD, no SI, and it becomes sinister snail. So it is no exception. So it was really single gene locus. This 100 determinant gene must be. So this was the one result. So this come out in the front page, and also this part, and this science wrote about it, so I'm quite happy about it. But then I got a strange idea again. If that rotation, that the third cleavage is so important, what happens if I push it by hand and a microscope? That's sort of crazy idea, but it worked. But they had a terrible time to persuade my group of people. Students laughed at me, postdocs laughed at me, and I managed to persuade very good technicians to do the work, maybe after half a year. So this is what I've done. This, it's easier to do, operate, manipulate the left-handed one. So 4-cell stage, start pushing it under the microscope until cell-cell contact is established, maybe half an hour. And you cultivate this, culture that. And for dexter one, it's a bit difficult because there's SD and SI, chiralitis starts much earlier, but you start push it a bit earlier and manage to do it. What happens? This is how we did it under this sort of microscope, and this is the result. This is the trochophore stage, this is the Berger stage, this is a juvenile one, and this is adult one. You from different sides, and also you take the shell off to see the internal organs. Okay? And this is the dexter snail, but we sinisterlize it at the very early embryonic stage. Look at this. Just like sinister snail. Bottom is the control for sinister snails, and this is the control for dexter snail. And if you do the dexterized sinister one, this was a sinister embryo, but you get just nicely, perfectly sinister snail you can get. So you can create mirror image creature. Then what happened the next generation? This is the ordinary one, sinister juvenile, sinister adult, and it produces sinister offspring, as because it's dictated by the gene in chromosome. Now, what happens if you manipulate it at the third cleavage stage? You get the dexterized juvenile snail, you get the dexterized sinister snail that is complete mirror image shape, but offspring reverts to the sinister because we didn't do anything to the gene. Okay? We didn't tap the gene, we didn't do anything to the DNA, but we did just pushed cells at the earliest stage. But so that means what we did is what the gene is doing, but in the opposite direction. So then how can it be? This is the case in mice. Mice, 7.5 days, you will start to see this nodal flow. This is the cilia, tilted and rotates, produce some flow, and that determines left-hand side. That's 8.5 days after birth. Lefty and then PTX, that determines the gut coiling or heart. That is mice case. And there are in many, this works. And if you change the flow, it seems to be, it's changing the shape. And this is the mouse, chick, xenophas, zebrafish. Most of them has this cilia, nodal flow rotation. But it's different in snails because it determined a much, much earlier stage. And we checked it, the nodal, yes. The nodal is Demonere is here, NODA is here as well, and PTX also it's conserved. So we clone the gene, NODA and PTX for the Demonere stagnalis, and this is the wish. This wish means where it actually, the gene is expressed. And not 8.5 days. At the 49 cell stage, it's already expressed. And we work hard to put left-handed embryo and the right-handed embryo together to make a shot, photograph. Otherwise, people think you just flip the other one to create a mirror image one. So we have to put together. But now you can see it beautifully. Left-handed one, sinistral, and the dextral one, it's all on the mirror image side. The nodal gene expressed. You were just looking at from a different one. And PTX, slightly later, but already at 64 cell stage, it is expressed. Then what happens if you, this is much later stage, it's expressed, mirror image. What happens if you do the manipulation? This is the important one, so they have to give you wake up, wake up. This is the thing you have to watch, okay? This is the 49 cell and the reverse one. Look at that. This is the dextral one, but it's not like dextral. It's just like sinistral. So if you push the thing at the very early stage, the nodal expression becomes at the reverse side, mirror image side. That's the reason it works like this. Okay, so this is the trochophore stage, the same. If you manipulate it, it's expressed in others. So it's just like this. Okay, so even at 2 cell to 4 cell stage, it doesn't work. It just goes back to the original chirality. But if you do this, you can change the gene expression site, and you can end up with beautiful, healthy mirror image creatures, which can produce offspring. But it goes back to the chirality that is dictated by the gene. So that is one summary, and then, that's coming out Nature, etc., etc. Then I thought, well, I'm working on Demonere stagnalis. That is 98% is dextral, 2% sinistral. That means there may be some potential. You can flip, change the chirality, because within a species you have both handedness. So I did the same work for Pfizer acuta, only sinistral only. Can we do the same thing? And indeed, we could do the same thing, and we could create first the right-handed Pfizer acuta. And if you look at the offspring, there is no exception. We worked on 248 eggs, all become going back to sinistral one, just like control. So it goes back to the one dictated by the gene, but the same mechanism operates for Pfizer acuta, for Demonere, or some other gastropod. Then we've been working very hard to identify the gene. Actually, it's really hard, maybe 20 years or so, and that we reported it. And what we did, make a long story short, we did the single-handed dominant gene locus was mapped by genetic linkage analysis, back cloning, and chromosomal walking using pure DD and pure snails, and also F2 to F10. We had the backcross lines. And shotgun sequences, the 7 back clones were assembled into contigs. Actually, it's just a bit of trouble to make it one contig. And also the physical map by alignment with AFLP linkage markers. And then quantitative real-time PCR analysis showed 13 out of 15 candidate genes act with maternal one. We have to work on maternal one. Then out of 13, 5 are actin. We know it's something to do with actin. So we picked up 5 actin-related genes. And we did the 3', 5' race. And this is what it is. Actually, we're not lucky that we had 780 kilobase. There's no recombination. So cool spot for the recombination, cold spot. Then out of that, there's a 15, and we picked up 13, maternal one, and we sequenced all And this diaphanous, there's a diaphanous 1, diaphanous 2, they are tandemly repeated. Diaphanous 1, you can see the left-hand hand, so one base is missing. That's the reason. You can see it like this. The dexter snails had the diaphanous 1, diaphanous 2, and 1 and 2, 89.4% homology. It's very similar. But sinister 1, dear 1, it's truncated. One missing C, it's coming to terminus. So it just stops there. So it's the same diaphanous 2. So this makes the difference for the left-handed and right-handed coding snails. And we did the quantitative RT-PCR, and you can see it's only for the dear 1 is only for the dexter 1, and for the sinister 1, and there are 2 in both, but it drops very sharply. And this is again to see where the gene is expressed. And although dear 1, the dear 2 is very, very similar in the DNA sequence, we managed to discriminate these 2. And this is the first cell before the first body explosion. This is also first cell. Then after that, it drops. But the harder N for the sinister 1, for the LS DR2, LS means diminished analysis, by the way. DR2, there are some, but then it drops. But there's no localization. That's the important part. Unfortunately, there's a sad story. You were writing this paper to Nature, and some paper came out. This is completely wrong, so it's really embarrassing. They couldn't discriminate DR1 and DR2, and they published these results saying this delocalization is the cause of chirality. This is completely wrong. We have to repeat the way of experiment, and there's nothing like that. And also some independent one from Princeton wrote to me, they repeated it, agrees with ours, or some very established one also congratulate on your true work, so I'm very happy about it. Anyway, we did the Western blotting, made the antibody, and again, now nothing in the first cell. So it's translated already at the oocyte, then it produces, that is maternal one. But there's some for the dexter one, and if you remember FD congenic one, because there's nothing for the DD and FD10, F10D, there's one. So it's something like that. Okay, time is coming. So I have to tell you this. For the human being, we had a situs inverters. There are people like that. So for the vertebrates, the CDS starts to rotate 7.5 days, and you see the nodal expression 8.25 days, very transiently it appears. But for my case, look at this. 3 hours at 8 cell stage, chirality is determined. And 24 cell stage, there is an organizer series already established. Then nodal appears 49 PTX a bit later. So I think it is really ideal to work on this snail for understanding the chiromorphogenesis. And this is one very good technicians. I didn't normally have a very big research group, but thanks to JST, Erato, I could employ postdocs and could work. But he's my PhD student who worked on this left-right mirror image doesn't exist. It's him. Masanori Abe, he's still with me and really my second hand. He didn't want to go to PhD course. He wanted to become medical doctors, but he was doing backcloning, very good work he did. And of course, future, I've done a CRISPR-Cas, so I know that is a gene. So you have to wait until I submit the paper, start writing. And I want to see the role of difference, 89.4% homology. What's the difference? What makes difference? And also there's the evolutionary event, and also how the chirality information transferred from 8 cell stage to 24 cell stage. That stage, organized cell is established already, so that is fascinating. So I talked about this, Alzheimer's, and you don't think there is no connection between the two, don't you think? But there is one connection. This is a 2000 scientific report, effects of A-beta exposure on long-term associative memory and its neural mechanism in a defined neural network. And if you expand it a bit, wow, they're using linear stagnites for the behaviorological work. I just couldn't believe it, but that's the connection between the two topics I told you today, all right? So I thank you for your attention, and you may, after another talk, you get a nice cup of coffee, and this is the Italians think, give me a cup of coffee, but there's a deal here. Okay, thank you for your attention.